Hello, this is David. This clip is not a teaching clip. The purpose of this clip is to ask a single question. And the question is, what determines the contour and shape of the maternal intervillous space of the placenta? Now, Dr. Burton's work and writing, which is very brilliant, often refers how you have your inner cell mass right here, your inner cell mass, and then you have initially this outer cell mass, which is sort of like your syncytiotrophoblastic and cytotrophoblastic, layer, and then initially it surrounds the entire amnion. And then what happens is there's this remodeling phenomenon that creates this discoid placenta that's only a disc in a single location. And so right here, this portion, the discoid placenta, and that it's sort of that the placenta, that the villi disappear from other parts, that they disappear from here, where I'm drawing the arrows, and that they really are the discoid placenta only occupies a portion of the sphere, and that's usually the deeper portion, the portion that's facing the myometrium, the portion of the original implantation that's facing the myometrium. And if you read that literature, you get the sense that you have this sort of mass of syncytio and cytotrophoblast, and initially, it has these hole-like spaces, which are potentially the glands. And initially, the fetus, and I just draw the fetus here with a circle. Initially, the fetus and the inner cell mass gets nourishment, not from maternal blood, but actually from maternal glands. And I guess if we take that model quite literally, you could sort of imagine that ultimately what becomes established is this sort of mass of syncytio and cytotrophoblast sort of has like a Swiss cheese appearance to it. It sort of has holes. And eventually it establishes an interface with the mother, which I'm showing here in yellow, and it invites in maternal blood to come in and sort of bathe the villi, and that's what creates what we call the hematochorial relationship, the relationship between blood and chorion. And this interface, which I'm showing in yellow, is the interface at which the maternal blood is invited to come in. But one reading of this model would say that here what I'm showing in yellow is a boundary. And the actual component of the intervillous space of the placenta, even though it's maternal blood, the contour is configured by the pre-existing holes and spaces, by the pre-existing sort of porous shape of this pre-existing syncytiotrophoblastic Anlage, if you like. That last statement is counterintuitive to what I usually see histologically. And let me explain what I mean. My sense isn't that the placenta itself determines the contour or all the constraints of the contour of the maternal space. Why? Because imagine another model. Let's say we take another model, and this is, let's say, your inner cell mass. Here we could draw the fetus here for the sake of argument. And here I'm drawing the discoid placenta, and let's ignore the problem of the regression of the placenta from this component. Let's just ignore that. And let's just imagine that here you have these remodeled maternal spiral arteries that I've shown in green. And there are these massively expanding, don't look at this part, don't look on this side of the yellow line, just stay here for now. And so you have these remodeled spiral arteries that I'm showing in green. And the model that I'm sort of espousing here is that the placental villi grow into these remodeled spiral arteries. And here you can see the deeper spiral arteries that are not yet remodeled. And somewhere here will be the actual, interface, the maternal-fetal interface. I mean, all of this is really the interface, but at some point, the placenta has to stop. And, these intraendometrial and even intramyometrial remodeled spiral arteries that, yes, they are remodeled by the extravillous trophoblast, here I'm drawing some more of them in green, have this pre-existing sort of plexiform architecture. And the villi, these villi, here I'll put a yellow arrow, these villi grow into these pre-existing spiral arteries. Here they're growing in, and somehow that space becomes confluent, But therefore, the pre-existing scaffold of the maternal spiral arteries, shown in green, somehow imposes a constraint on the topology or geometry of the maternal aspect of the circulation. Because you could imagine, let's say right here, this is your basic villus growth pattern as a tree, right? So your villus have this certain intrinsic biological program to grow as a tree. And here I'm just showing obviously like a two-dimensional section of a tree. The whole time I want you to bear in mind that imagine this is a real-life tree and you're taking a single two-dimensional plane. So the pathologist is encumbered by this because we're only looking at two-dimensional sections. But on the two-dimensional slide, we see sort of this section of a tree that looks like this. And imagine that this is the internal drive of how villi like to grow. But if the villi are growing into spaces with various shapes, the shapes, so imagine right here again, right here, that this is sort of the native shape that the villi want to take, with a central stem villus and then smaller and smaller sort of leaves, smaller and smaller exchange and terminal villi growing around, sort of like Medusa's head. But if you put that into a pre-existing container, i.e. the spiral artery, it imposes a shear, it imposes a stretch, it changes the function. And so it imposes a stretch on the function or a shear on the function, and so you imagine you put this in here and it changes the shape somewhat. And therefore, it's sort of like it puts a reflection on the image or a rotational vector on the image. And so you can imagine you have this pre-existing villus constraint, but by growing into the topology of the bays and gulfs of the maternal plexiform spiral arteries, you impose some sort of change on the shape. So you go, let's say, from a shape that looks like this here to a shape that looks more like this, because it has to conform to the contour. And ultimately, how the entire maternal hematochorial space, which I'm sort of showing here, here's like a model of the placenta, becomes confluent, I'm not exactly sure. But there are clues that what I'm saying has some validity, because like when you look at the placenta, you have your branching tree. Imagine here in blue are the chorionic arteries of the baby. These are the branching villi seen on a two-dimensional section. You sometimes see these stem villi, and my yellow is my extravillus trophoblast, and you see them within the placenta in islands and columns of fibrin that sometimes articulate with the basal plate, and sometimes, as I'm showing here, you even see some free ones, and that's interesting. And then sometimes you even see septa within the placental matter, if you catch a good section, of columns running up with extravillus trophoblast and fibrin, and villi do anchor into it, stem villi, and sometimes they anchor in like this, like sort of perpendicular or parallel actually to the plane of the column, but sometimes they come in curved or even perpendicular. And that to me suggests that this is somehow telling me the tale of pre-existing, somehow this relates to the pre-existing spiral arteries that the extravillus trophoblast came into and remodeled. I don't have the full solution, but when you look at the implantation site, just to review, you have extravillus trophoblast, within fibrin, and then you have the decidua, and you have these big spiral arteries, and sometimes you even see gaps like what I'm drawing here in the fibrin, and you could imagine that the negative space of that could be a spiral artery here at the bottom, what I'm showing in yellow, that the blue line here is the plane of cleavage, where the placenta separated when it came out, the blue line here. I'm going to turn some of the blue into yellow just to make clear where I'm at. But sometimes you see, when you get the placental specimen as a pathologist, you see a gap in the fibrin. Look carefully the next time. You see a gap, and then you see villi growing into that gap. And sometimes you might say to yourself, well, that's just an artifact, or they're just misplaced villi. But if you've ever looked at a placenta accreta, you would say, no, it's probably not. It's probably very deliberate. And what I think it is that there's some continuity between the fibrin here and a remodeled spiral artery that's just outside the plane of cleavage, and these villi are growing into this spiral arterial space. And so because we're actually catching villi growing into the spiral artery, that tells me that the growth pattern involved exactly that, villi growing into spiral arteries, and there's some kind of an interface here where the intraplacental component, the spiral artery, becomes sort of extravillus trophoblast, or even the syncytiotrophoblast lining here, and these columns of fibrin and extravillus trophoblast, whereas on the maternal side you have like these remodeled spiral arteries, which are spiral arteries, and then with your extravillus trophoblast that have of course taken over the lining. It's no longer maternal endothelial cells. And so this, when you look at the basal plate of a placenta, you see these gaps with these villi growing in, and that's not accidental. And the other thing that you sometimes see is these discontinuous columns of extravillous trophoblast and fibrin. And then you see, sometimes you really do see septa. You'll catch a septa like this one growing up at 90 degrees for a variable portion of the height of the placenta. And I just imagine what would happen if somebody would use stacking and mathematical modeling and reconstruct all of these discontinuous, islands and septa of villi and not of villi, septa of extravillous trophoblast and fibrin. I wonder if that contour would somehow hearken to the contour of the plexiform maternal vessels. Now, the best place to look at the true contour of the maternal plexiform vessels is not in the decidua basalis, but in the decidua capsularis, because there the villi didn't grow in, and you get a sense of their contour, and what you see is that they look like this true plexus. Now, you might be saying to yourself, David, but this plexus is so small. How could you go from a structure like this plexus that I'm showing in green that we see on the decidua capsularis, how could you go from that structure to this confluent open space, even if I allow that there is some septation caused by these extravillous trophoblast and fibrin? And my answer is, maybe all of this, now I'm going to say something subversive, maybe all of this is a single remodeled, is the continuity of a single remodeled spiral artery. One. And you may be saying to yourself, that's totally crazy. That's a huge scaling up. And I would answer to you, so is pregnancy. Look at the size of the non-gravid uterus and look at the size of the gravid uterus, and there's your clue. It's a massive scaling up. It's not a minimal scaling up. Your non-gravid uterus to your gravid uterus is a massive scaling. And so I would imagine that the end fate of the spiral arteries, when it's expanded by the ingrowth of villi, is scaled up, not minimally, but massively. And the last point that I just make is, there are some people that have said, like when you look at a placenta, like a cross-section of a growth placenta, you sometimes can see, the remnants of entry of where the spiral arteries come in, and you also sometimes sort of get a sense that there's like little gaps and stuff like that. And of course, Dr. Henning Schneider was actually able to cannulate these, which is unbelievably amazing. So, I guess the point of all of this, my question is when we look at the contour of this maternal space here, with its sort of, ebbs and eddies, because we want to ultimately model flow in this maternal space, right? And when you look at the contour of this maternal space, it doesn't look like the outside of a branching tree. It just doesn't. Look at it yourselves on the slide. It has a lot of curves. It looks like a paisley tie. It's a very curvy space. It has this intrinsic curvy contour to it. And I wonder if what we're seeing in the contour of the maternal space is somehow the residue of remodeled confluent maternal spiral arteries that were initially very, very small and plexiform, like what we see in the capsularis, and have now been conjoined. And then what we see, in the basal plate and the maternal side are some pre-existing, quote-unquote, uninvaded, arterial spaces. And I would really want to get people's feedback on this. This is a question that's haunting me, but I think it's really important. Because and this is why it's so important, if we could argue that there are certain geometric constraints on the topology of the maternal space that are imposed by the pre-existing plexiform nature of the vessels in the endometrium, in the decidua basalis, then we understand some of the mathematical constraints of this space, of this whole maternal space. And we really want to understand the mathematics of this space because we need to start modeling the flow vectors. And that'll help us better understand disease. So I'd really, really love to hear your thoughts on this. Thank you so much.